we have a fun afternoon planned. Class and lab. Happy Tuesday. Ready to learn all about the taste system? All right. Any questions about olfaction? No, we are all good to go on sense of smell. Pheromones, fungal nasal organs, and all that jazz. Good. All right. So, back to the chemical senses. That's what we're talking about today. And so I think I mentioned this last time that we sort of know the least amount about olfaction and taste. And that's mainly due to quality of life issues in terms of other sensory systems, right? So if you can't hear, that's a bigger, or can't see, like those deficits are bigger quality of life issues than you can't taste or you can't smell. And so a lot of research has been devoted into understanding the vision and the auditory system in order to come up with better treatments and to try and understand these deficits and things like that, right? Also, with the chemical senses, both of them, smell we talked about last time has the ability to regenerate and repair itself after damage, particularly the peripheral areas. So damage to those areas isn't typically permanent, right? And we'll see the same thing with the taste system. Taste cells turn over about every 10 to 14 days on your tongue. This is also one of the things that really drew me to become a gustatory researcher, to study taste as my research area, is because there's a lot of big questions left to answer. There's a lot of things we don't understand that we're still trying to figure out. So it's a big, wide open space to sort of explore in terms of science and research. I would argue, and I made this point last time, that the chemical senses, probably from an evolutionary perspective, are some of the most important, right? And you look at other species, and they have invested a lot more resources in smell than we do, and in taste. Vision, all the other senses, right, are activated through some intermediary receptor, right? Rods and cones, hair cells, mechanoreceptor structures, things like that, right? Where taste and olfaction actually interact, the neurons interact with the physical stimulus itself. All right, so what is the job of smell and taste? Well, they're like the gatekeepers, right? The protectors of what you put inside of your body, right? Things only get inside of your body two ways, through your nose or through your mouth. And so smell and taste are looking at what molecules are coming into your body and trying to protect you from harmful ones, and trying to encourage you to consume good ones, right? So we have this sort of monitoring system to identify good things, particularly with taste, right? To identify good things and promote consumption of those good things. What are some good things? Food? What kind of food? I don't know what tastes good. Fruit. Fruit? Someone said chocolate. Right? So,
cooked things rather than raw.
yeah, cooked things rather than raw, for some, right? So, I want to keep interacting today. I get some look, I take these notes. So, why do we eat those foods? Why are they good? Yeah, they have things our bodies need and can process, right? We need salt, we need sugar, right? We need energy, we need fuel, we need vitamins, we need all kinds of nutrients. And those are the things that taste good, right? There are bad things that we don't want to consume. What's something bad tasting that you don't want to consume? Sour milk, right? Rotten milk, right? And we call rotten milk sour milk because it has a sour taste, and things that are sour, that are spoiled, in general, we identify those as not tasting good, and we have an innate desire not to swallow that, right? Not to consume that thing, right? We don't even want to smell it. And everybody's like, here, smell the smoke. It's bad, right? You don't even want to do that. All right, so, and as we talked about last time with the olfactory system, these two things are not independent of each other. They actually work very closely. We're going to demonstrate that in lab today and look exactly at the role of smell in flavor production, right? So when you eat something, you say that tastes good, right? You talk about the taste of that thing. And in your mind, you're thinking, hey, that's like what's coming out of my tongue, right? That's the sense that's coming out of my tongue. But really, what you're doing is talking about the flavor, right? It's very hard to isolate just the signals. When I talk about taste, as a researcher, I'm talking about signals that come from your tongue and go up through the gustatory system to your brain, right? But those get combined with smell, with olfaction, and with touch, mouthfeel, and with vision, even, in the orbitofrontal cortex, and that's our overall perception of what we ate. And unless you're doing it blind, without any smell, and without any texture, you're not really just looking at taste, you're looking at flavor. All right. So, very special role of taste with digestion, and there's interactions both ways, bidirectional with the hypothalamus. And we'll talk about that a little bit next time. There's also special interactions with emotion and memory. So, and from an evolutionary perspective, when you ate something that had high calories or your body was craving, right, you would want to form a memory, just like we talked about with smell being protected. Here, we want to form a memory of how did I encounter that food, where was it, what was it like. I want to associate a positive emotion that's going to motivate me in the future to do that same behavior, to get that same access to that food, right? So there's strong connections with things like the amygdala. Most food has some hedonic value. You talk about hedonic value, that's the emotional value, the positive hedonic value or negative hedonic value. Memories with the hippocampus and mood, emotional state, right? When you're feeling down, right, and you go and you get your favorite ice cream and you eat all of that favorite ice cream, how does it make you feel? Nobody does that, just me. Does anybody have a emotionally comforting food? Right? We talk about comfort food, things that makes us feel good when we eat it, right? Because we have that strong connection with the emotional system, right? And conversely, I'll say if you probably took a big swig of sour milk and didn't realize it, you probably had a negative emotional state after that, at least for a little while, right? All right. So, because like olfactory receptors, our taste bud receptors interact with the environment, it makes them susceptible to being damaged. And because of that, the system is very adaptive in being able to regenerate, have neurogenesis, to create new taste receptor cells. And actually, in the taste system, unlike olfaction, so olfaction can repair from damage, right? The taste system, unlike that, undergoes this regular neurogenesis, right? So every 10 to 14 days, we are creating new taste bud receptor cells on our tongue. So right now, the taste receptor cells that are on your tongue today weren't there when we started this class in the fall. Yes?
Sorry, my dad had throat cancer like a year ago. Like a long period of time, I never tasted like trash because of is there like something that can keep that from happening then? He was having taste issues.
Yeah, so sometimes that chemotherapy will damage the taste receptor cells. And so, but when it's over, right, when that treatment's over, they should recover and the sensitization should return because new taste cells will come in that aren't damaged to replace the ones that have been exposed. All right, so already gone over this. Flavor is combined, many senses. We're talking about taste, we're talking about sensations coming from the tongue. So what, unlike olfaction, it doesn't have primary sort of qualities, right? Olfaction's all about memories and objects, which was identifying it. With taste, we can break things down into basic qualities, right? So what are some of those basic qualities? Categories. Salty, sweet, umami, good. What? Sour. That's umami. One more. Bitter. Thank you. Right? So why sour, salt, sweet, and bitter? Those were the first four, right? Then came umami. Now there's a bunch of us researchers that are proposing that dietary fat has a taste. There's a taste for fat. It's been coined. Oleogustus is the name for the taste of oils, if you will, right? But those four, why those four primary taste qualities emerge as important? You know? Yes?
I guess just like for sweetness, our body needs glucose and like our body also needs salt.
Yep.
And then bitter, I guess, helps us determine things that may not be good for us.
Yeah, absolutely, that is right. So there's evolutionary pressures for those four categories, right? The earliest one was salt. And so the earliest life forms on the earth were found where? In the ocean, right? They were aquatic, right? And so in the ocean, aquatic animals actually express taste buds on the cell's surface, on the surface of their body. And they're detecting what is the salinity, what is the amount of salt that they are in, right? So you have freshwater species that don't like to be in salty water, and you have saltwater species that like to be in the ocean and salty water, and they have to know, hey, what environment am I in? What is the environment like, right? So that was probably the first taste that emerged, and it was really not a taste as much as it was an environmental detector, right? Sour also was a safety mechanism, right? So when something's decomposing underwater, it's giving off hydrogen molecules as it rots and decomposes, and that is what hydrogen is the taste of sour, right? The acidic taste. So an acidic environment is not a good environment in the ocean. And so again, these two were related to sort of aquatic environmental detectors. Then and fish don't have receptors for sweet and bitter. They do have receptors for salt and sour. And then as animals evolved on land and became terrestrial, and we had to seek food sources, right? Two more tastes evolved. Bitter as a toxin indicator, so most natural poisons activate our bitter taste receptors and initiate an innate rejection response. And sweet for nutritional value. And it just so happened that we had the holdover from salt that's also an important thing, so that became an appetitive stimulus as well. All right, so looking beyond those, somebody mentioned umami. Umami is savory, is the word savory in Japanese, and it is associated with amino acids, so the meats, broths, monosodium glutamate is a classic chemical that stimulates or creates the umami taste, right? It was first described in the early 1900s, but was large, and it was described in like Japanese culinary practices and used widely in those cultures. But it wasn't until the 1980s that researchers discovered the transduction mechanism in the taste buds for glutamate and these amino acids. And then we're able to identify neural signals and map out the entire sort of system and show that, yes, this is a functional component of the taste system. It was in 2000 that they identified the transduction mechanism for MSG. And so I'll talk next week about the work in my lab and Tim Gilbertson's lab and Rick Mattis' labs, a couple other people that have begun exploring whether or not dietary fats produce a taste. If you ask somebody, why do you like to eat fatty foods if you know that it's not good for you? And it's because it tastes good, right? French fries taste good. But they don't taste good until you put in that fat, right, in that oil. So I'll talk a little bit about that and about that story. All right. How many of you have seen a picture like this of the tongue and how taste is represented on the tongue, right? Seen this before, high school maybe, something like that, right? It's totally wrong. So take that out of your head. That's not the way the tongue works, right? All parts of the tongue respond to bitter, right? All parts of the tongue respond to sweet. All parts of the tongue respond to salt and sour. They all respond to everything, okay? Now, do they have different intensities? Yes, right? Are some parts of the tongue more easily stimulated by taste? Yes, and that's sort of where this overly simplified tongue map came from, right? So the front of the tongue responds to sweet, salt, sour, and bitter. It responds least to bitter. The size of the circle shows how responsive it is, right? It responds least to bitter and most to sweet and salt. And the back of the tongue responds most to, responds most to sour and bitter and least to sweet and salt, but it's responsive. You get those responses there. They are taste buds. Taste receptor cells in those taste buds for all of the taste senses. So there are three types of papillae, and the papillae contain the taste buds. And so three types of taste papillae. There's fungiform papillae, folate papillae on the side, and circumvallate papillae in the back. So fungiform is anterior, folate's on the side, lateral, and circumvallate's in the back of the tongue. And as I mentioned, they're equally responsive to all taste classes. Really, maybe not equally responsive to all. They're responsive to all four taste categories. They're not equally responsive because they have different sensitivities, right? Some of the areas respond more to sweet or more to sour. All right, so if I take this part of the tongue, right, and I take a picture of it, right, then I can, and I stain it with some blue food coloring, take a picture of it, we can see those fungiform papillae. They're these big things. The little things are touch papillae, filiform papillae that are innervated not by taste neurons, but innervated by mechanoreceptor and free nerve endings and give us our texture and our temperature of taste in the form of the tongue. So normally in this lab today, I'd be taking pictures of all of your tongues and we'd be counting your fungiform papillae, right? But because of COVID, you're going to get out of that because we're all supposed to be wearing masks and I don't want to act like a dentist and put it in your mouth and take a picture of your tongue. But this is a picture of a former student's tongue, and this is what it would look like. And we would actually be able to count those fungiform papillae. We're just not going to do that in the lab. We have data from other universities we can use for that. So if I were to take that and take a slice of your tongue, which I don't do to my students' tongues, this would be the top of the fungiform papillae. And the sides around here have these trenches on either side, and then in the trenches, these are actually taste buds here, right? These are collections of those taste receptor cells. about 50 cells in each taste bud. All right, so here's a taste bud that's been blown up. Here's a cartoon of it so you can kind of see it a little bit better. But it has almost like an onion-like appearance. Like I said, there's about 50 cells. This is what we call the apical side, where the cells can interact with the taste buds. And then we have the basolateral side, where there are nerve fibers connecting to those taste buds. As I mentioned, they regenerate every 7 to 14 days. That's one of the things that we don't really understand. Because if you think about it, if these cells are constantly dying and a new one's coming and taking its place, you think that that would affect our ability to taste, or our sense of taste would change as these things are changing on our tongue. Yet it stays really constant. Right? And so we don't even, I don't think we even understand if this cell primarily was responding to, say, sweets, and it dies, does another sweet cell come in here and hook up to this fiber? It would have to in order to maintain our sense of, our common sense there. So, but we don't understand exactly how that happens in the tongue. David?
Are taste buds and papillae the same thing?
Taste buds and what?
Papillae?
The taste buds are in the papillae, right? So here are the papillae, right? This is a papillae here, and these are taste buds. Each one of those little dots contains 50 cells or so, taste receptor cells. All right. So a little bit about this. So when you eat food, right, and you chew it up, you're releasing chemicals into your saliva, into solution, and those chemicals travel into those pits, those grooves, and interact with the apical side of the cell, and get transduced into a signal that is then going to activate that taste bud and release neurotransmitter on one of the 3 afferent gustatory nerves. Cool? All right, doing bottom-up signal. Yeah. All right, so there's this apical basolateral distinction, and in between those is a thing called the tight junction. And the tight junction is just what the name says. It's like a barrier or a wall. It's going to keep salt or sweet molecules from coming down and interfering with this, particularly salt, right? Because down here, we're maintaining the salt balance so that we can get depolarization and we can get all the things that a working cell needs to be able to do. So this area is exposed to taste buds, and they stay in this area, and then this is sort of the working area of those cells. Yes?
So would it be possible to have too much salt to the point that it affects like your ability to like the sodium channels, because it's like NaCl that is
No, you can upset your osmotic balance by consuming too much salt to cause serious problems. In fact, that's how we kill people with lethal injection. We give them a high injection of potassium, and it stops all the nerve signal in the body. All right, don't want to do that. Don't play around with your electrolytes. Your body will do a lot of stuff to try and balance that out before it gets to that stage, right, to regulate that. All right, but I'll take the opportunity to mention that when you have hypovolemic thirst, which is thirst because you're losing volume, but you're also losing salts, that can initiate a salt appetite. So again, not just the digestive system interacting with the taste system, but we can also have our thirst system interact with taste to stimulate a craving for salts when salt is too low. Right? So you consume too much water. All right, taste receptor cells, synaptic contact down here with our afferent sensory nerves. All good. Oh yeah, and so then we also have these basal cells. These are the cells that are going to develop into new functioning taste receptor cells. We call them basal cells. All right. Okay, so one of the things that we've just recently learned is that only certain cells actually form synaptic connections. And there are some taste receptor cells that don't have a connection with an afferent sensory nerve. And for a long time we didn't understand, like, what are they doing? Are they just kind of waiting in the wings to be used? Or, you know, are they functional? And it turns out that within just like the last 5 years, we discovered that these cells are actually communicating with one another. And this cell, for instance, that's not hooked up to a nerve, might communicate to this cell and cause it to be depolarized and send a signal. And that communication happens through ATP molecules, we think. And so the other thing that we don't fully understand is, unlike the olfactory system, where one olfactory receptor only expresses one type of metabotropic receptor, here taste cells express multiple transduction mechanisms, but they don't necessarily express all of them. And we don't exactly know why cells might be a salt and sour cell, or just a bitter cell, or a bitter salt and sour cell. We don't exactly understand how that gets arranged. All right, so if I take a cell and I put my tight junctions in there, so this is my apical side, up here is the lateral side. The basic transduction is either through ion channels or through metabotropic receptors, lock and key mechanisms, releasing a second messenger G protein. We're going to get depolarization inside that cell. And again, like I mentioned, we have a balance of salts down here on the basolateral side to maintain those potentials. But when we get depolarization, we're going to release calcium either from an internal storage locker, or it's going to come in through a voltage-gated calcium channel. And calcium then is a signal for exocytosis, just like in a neuron, and we get neurotransmitter release on the neurons. Again, things we don't fully understand. What is the neurotransmitter here? What is the signal? And it looks like there's evidence for at least 3 different types of neurotransmitters that are involved in signaling from taste receptor cells to the afferent nerves: serotonin, norepinephrine, and glutamate. Why have 3? Why not just have 1? We don't know. We don't fully understand that, right? And in fact, some of this has come from research of people that are on antidepressant medications that increase serotonin, and the fact that caused changes in taste perception because it was influencing serotonin in the taste system as opposed to in the limbic emotional system. All right, and then we get an action potential in the nerve, and that's going to go up to the nerve. So the ionotropic transduction, that's for our sours and for our salts. Sours and salts use ion channels, right? Salts can simply come in through a sodium channel, just like if you were doing an EPSP in a neuron, and depolarize the inside of the taste receptor cell. Sours are typically hydrogen proteins, hydrogen molecules, and those hydrogen molecules will block, they can sit and block a potassium channel that's normally letting potassium out of the cell. And if we block that and trap the potassium inside, that's going to make it more positive and cause depolarization. Good? All right. This is another example. It's a little bit more detailed, right, where we see the primary way. Hydrogen, there are some channels that allow hydrogen to come in to the cell. But primarily, we're just going to talk about sours. Hydrogen blocks that potassium channel and traps potassium inside. Salts come in and depolarize. Everything else is metabotropic. So bitter, sweet, and umami all are metabotropic lock and key mechanisms. Fats, when I talk about them, I'm not going to talk about them today. When I talk about them, we're going to see that there's both ionotropic abilities and metabotropic abilities for fats. They use both. All right, so Just like a regular leptotropic channel, sugar molecule sits and activates it. Second messenger is released. That second messenger causes calcium release inside the cell and get exocytosis. All right. So bitter, sweet, and umami all act through that way. A little bit more about those, right? And again, like there's some things like you'll know there's some missing transduction mechanism here in the umami that we haven't quite figured out yet. So again, lots of things to be explored in the taste system. All right. Bitter, looking at bitter molecules. So the bitter receptor family is named the T2R and then some number, right? So it would be like T2R1, T2R3, T2R13, etc., right? And we know that there's about 30 to 40 different types of bitter receptors, right? All of them produce that bitter taste, but they bind to different chemicals, right? So here we see, we'll be testing PROTE today. So PROTE interacts with 4 and 38. So if you're a non-taster today and you can't taste that PROTE, you have a genetic allele that does not make receptor 4 subtype and does not make receptor 38 subtype. That's what allows you not to be able to taste PROTE sensitivity. So we have different genes that are coding for each of these sort of variants that are binding with different bitter chemicals. Cool? Good? All right. With sweet receptors, they belong to the T1R2 and T1R3 families. There are sort of 2 binding sites, and we can have small sweet molecules that bind to one part of that metatropic receptor, or larger sweet molecules that bind on the other side, or we can have what's called a heterodimer activation, right? Where you have a complex carbohydrate that's going to sit and interact with both of those receptor sites. So any one of those 3 conformations is going to stimulate the second messenger release inside the cell to get our sweet taste. Umami receptors are from the metabotropic glutamate receptor 4 family, and there's some evidence that they may also stimulate T1R1 and T1R3 as well. All right, yes?
This gene, is it naturally occurring? Like it is have they like synthesized it? Now they can think about, is like MSG is bad for you? Like is it?
Yeah, so monosodium glutamate exists in nature, right? But and that chemical is like a primary activator of the umami receptors, right? Just like sodium chloride is sort of the primary salt, although there's other like sodium carbonate, sodium glutamate, and a whole bunch of different salts, right? Including potassium chloride and calcium chloride that also taste salty, right? But that's sort of the prototypical one. So that's kind of the way you think about MSG. So the other things that activate the umami are amino acids, right, that are found in meat, and other things. But yes, so now we have the ability to synthesize MSG and add it as a flavoring, right? And some people are more sensitive to higher levels of MSG, right? And so when you put a lot of it into food, some people can respond negatively to that. I don't think it's necessarily dangerous, but some people respond negatively to that. All right. We're good on transduction, receptors, physical stimuli. All right, pathway. 3 nerves are going from the mouth to the central nervous system. So we have, and they're all cranial nerves. So we have cranial nerve 7, the facial nerve, is the chordae tympani nerve. It's a branch, it's just one part of cranial nerve 7. The chordae tympani nerve does the anterior 2/3 of our tongue. The glossopharyngeal nerve, it's cranial nerve 9, does the back 1/3 of the tongue. And then the vagus nerve innervates this sort of top part of our esophagus here, and that also contributes some taste there. Rats have another branch of the facial nerve that innervates the palate, the top of their mouth. Humans don't have it. All of these converge in an area of the brainstem called the NST, the nucleus of the solitary tract. And so they come in order, right? First the chordae tympani, then the glossopharyngeal, and the vagus nerve, and they're all found here. And this is also obviously happening on the other side. Sort of the NST kind of lies next to the fourth ventricle ahead of the ovax. All right, so they converge in orderly fashion. From the NST, the signal goes to several different places. It goes to an emotional circuit. It's kind of like olfaction. It gets distributed. So it goes to a emotional pathway through the parabrachial nucleus, the PBN, and to the amygdala. There's some debate about whether or not humans have a PBN. Rats do, monkeys do, but some debate about whether humans do. So in humans it may go straight to the amygdala, but we're going to talk about this as the pathway. The NST also goes through the thalamus, as all sensory systems do, through the VPM nucleus of the thalamus. And then from there it goes to primary cortex in the insular and frontal operculum. We'll mainly talk about the insular cortex as sort of being primary. And then from there it goes to secondary areas like the orbitofrontal cortex or frontal. All right. Pathway, good. Pathway's easy. Neural coding, okay? So if I put a stimulus on the tongue here and I'm recording from neurons in the rat that are going to the brain, and I put salt or sweet or sour or bitter or just water on the tongue, right? What does this neuron look like it's responding to? Sucrose, right? And is it really responding to any of the other stimuli? It's pretty much spontaneous activity there. So what kind of neuron would we say this is? Sugar neuron, which is what type of coding? We're talking about two general types of coding. In neural coding, what two types of coding do we talk about? Labeled line and a cross fiber pattern. What kind is this? It's labeled line for sweet. It is a sweet neuron. We call them S neurons or sweet specific neurons, right? What about this neuron? Now I'm putting salt, sour, bitter, sweet on this neuron. We're looking at its fiber pattern. Cross fiber pattern? It's not label line because it's not responding to only one thing, right? Label line means that this neuron that we're recording from here is active, sweet must be on the tongue, right? Activity equals sweet, right? Here, we're giving responsiveness to salt, sour, and bitter, not to sucrose, right? Can you see a difference in the pattern of action potentials for those three categories? Yeah, so this is like constant firing, but not intense firing, right? And for sour, we get constant firing that is much more intense. You think the brain could see this and discriminate it from this, right? And with bitter, we get this phasic on, off, sort of intermittent signal. So, you can visually look at that and identify those as three different things, and the brain can do the same thing. So it's looking at the pattern, the cross fiber pattern of those signals. All right, so if we record from a whole bunch, and this is actually data from my lab where we do neural recordings in rats and mice. So if we look at a whole bunch of neurons, here's 10 different neurons, and we see what is their responsiveness to sweet, salt, sour, and bitter. All of these pretty much only respond to sweet, right? Yeah. And so we get a whole bunch of these 1 through 10 that pretty much only respond to sweet, don't really respond to anything else, right? We keep going and we look at the responsiveness to 40 different neurons. We get a whole bunch that just respond to salt, and we get a bunch that are responsive to salt, sour, and bitter. This is a little bit better. This is, again, data out of our lab that shows what we call S units, units in yellow that are just responding to salt, N units, units that are in blue that are just responding to salt, pretty much. So we have 2 label blind signals, sweet and salt. And then everything else is a cross-fiber pattern, right? So here we have units that we name them based on what are they most responsive to, who they're most responsive to. So these are acid units because they're most responsive to acid, but they are a cross-fiber pattern because they're also firing to salt and to bitter. We also have some then that are most responsive to bitter, but also fire to salt and to sour. So we call these specialist and generalist neurons, right? Sucrose specialists, sodium specialists, and then we have neurons that respond best to salt, but also respond in a cross-fiber pattern to sour and bitter. Sour neurons that also respond to salt and bitter neurons that also respond to salt and bitter. So cross-fiber pattern generalist neurons. Hanging in there? All right, cool. All right, so for a long time there was a debate in the gustatory community about, like, it had to either be label line or had to be a cross-fiber pattern. But now, I think most people have accepted that there's roles for both label line and for a cross-fiber pattern in neural coding. And you could think about, you could theorize why that might be, right? What are the 2 label line categories? Tastings. What tastings use label line?
Sweet and salt.
Sweet and salt, right? Those are things we need that we're seeking in the environment, right? To have neurons dedicated to say, yes, you found sweet, or yes, you found salt, could be very beneficial, right? Label line to detect important nutrients that we need. Right? And then a cross-fiber pattern to discriminate between similar but different tastes. Right? And so, sodium chloride and potassium chloride, you can discriminate between those 2. And potassium chloride has a little bit of a sour component that the sodium chloride doesn't have because of the potassium ions interacting with that sour potassium channel. Right? So again, having neurons that fire to salt and sour allows us to discriminate without cross-fiber. And psychophysical tests tend to support this. So. All right. This is sort of how the pathway looks. There's a lot of bidirectional arrows. So the PBN is talking to the amygdala, and the amygdala is talking to the PBN. There's actually bidirectional arrows here. Well, here it is. It's coming in this way. The amygdala talks to the MST down here. Here's our gustatory cortex way up here. So this is sort of a more complex analysis. The hypothalamus is in there. But we're going to look at it sort of like this in this diagram. So I'm not going to make you learn that, but we'll look at this, right? Where we have these bidirectional inputs between emotion, digestion and hunger and satiety, the thalamus going to our taste, primary taste categories. All right. Got all that. Good? All right. Gustatory thresholds. So, most differences in in taste or in individual differences in taste are at that receptor level or at the transduction level. So like I said, super tasters and tasters and non-tasters of probe has to do with how many of those bitter receptors are in your tongue, are produced in your tongue. Right? There's some subtle differences between people that maybe they have more salt taste or more sweet taste in different locations on the tongue as well. If we look at changes in taste across age, we really, in controlled studies in animals, this is in rats as they age, and in humans too, the taste system really isn't affected. It stays pretty much intact. But if you talk to your grandparents, they'll probably and ask them about, has their taste perceptions changed? They'll say, yes, it has. But what really has happened is their olfactory system has become weakened and is not getting as much input. And so the flavor, the taste thing, is really flavor, and it's really a decline in olfaction and not a decline in taste. Right? So we look across these rats as they get really old. Really, the data is just through the 23 months, because after that, they really start dying and our sample size goes way down, so we get some weird things. But this is done by a friend of mine in grad school, Kurt Fall. He teaches at Millsaps now. But he had, this was his master's project, so he had to test these rats every 4 to 5 months for 2 years in order to get this one research project done, right? And we don't wait for them to age. It's just become old. All right. So, reduction in flavor perception due to decline in olfaction, not due to the changes in taste. All right. Thresholds, perceptions. So what can affect your taste perception? What are some things that affect the way foods taste?
Experience.
Experience?
Physical
Appearance? Okay.
Hunger,
Hunger? We'll talk about that.
if you're sick or hungry.
If you're sick or not, that's probably your olfaction, not taste, right? So if your nose is stopped up and you can't get those signals through, then you have a decline in taste perception and flavor perception, sure. What about the environment? Does that affect taste? Environment of your mouth? Right, so you have things like adaptation. What about temperature? How does cold coffee taste compared to hot? You have a hot cup of coffee and you're drinking it and you let it sit and it gets room temperature. Does it taste the same? No, so the temperature of foods can affect the taste as well, right? So there's lots of things that can affect taste. And whether or not fats are present can affect taste. So some of the evidence I'll talk about with like free fatty acids is that the fats act to enhance our normal taste. So, you know, if I gave you a plate of sucrose and a plate of Crisco, right, you probably wouldn't find either of those very appealing. But if you mix them together, you get frosting, which you probably would find very appealing. So there's some evidence that fat, like in Crisco, enhances that sweet taste and actually creates a new, unique taste. We can have adaptation, we can have short-term changes, right? And again, I got any black coffee drinkers in here, right? You drink black coffee and then you drink water, and how's that water taste? Different, right? Or if you're drinking orange juice and then you have some soda after that, have you ever done that, right? So these are examples of adaptation where you've adapted because of the presence of the coffee or the orange juice and change the sensitivity of those taste receptors on your tongue, and then you drink a different type of beverage and it initiates a taste, right? Conditioned taste aversions, experience, right, can play a long-term role on how we taste things. We already mentioned genetics. That's going to affect if you even generate a sensation, if you have a receptor, how many receptors you have to generate sensations. And then there can be cultural differences, and again, I think that gets into experience, right? If you grow up in India eating lots of curry and spicy foods like that, then your preferences for those types of foods are different than someone who's never experienced that sort of flavor, right? And so our experience influences what we prefer and won't prefer. And if you don't prefer something, like you didn't like coffee the first time you tried it, what do? You put a bunch of stuff you like in it, like milk and sugar, and then you begin to tolerate it, and eventually you might come to like that taste, right? So something that was initially aversive can become a competitive or a preference. Yes?
What signal, I guess, is triggered by spice?
Yeah, so chemicals in foods that are spicy are things like capsaicin, and they're interacting not necessarily, again, the same taste, not with the taste system. So spiciness is not a taste, it's actually a touch. They're interacting with those free nerve endings to make the heat, to make where it feels hot, right? So when you eat spicy food, your mouth feels hot, you feel like it's on fire, because it's that strongly of. We're always kind of adapted to salt, right? It's hard to produce a salty potentiation because our mouth is kind of constantly adapted to salt. All right, and so we have cross-adaptation. So because this is like receptor fatigue, right? If we have a similar taste, it'll also, and it's activating a similar receptor, it'll also share that adaptation, that cross-adaptation, right? And again, this is a clue to us that the adaptation is happening at the transduction level, like transduction fatigue at the receptor level. Yes?
Think about like things that cleanse your palate or neutralize other flavors. What category would they fall under? Or is it possible to actually like neutralize?
You're sort of like rinsing your mouth, like to get residual chemicals out, right? So, for instance, today in lab, I'm going to have you guys rate the intensity of different concentrations of sweet and salt solutions, right? If you don't sort of rinse your mouth out with some water in between each of those stimuli, you have some, like you just took a strong sweet stimulus. The next stimulus might be a weak sweet, right? If you still have some of that hanging out in those grooves and around the papillae, then it's going to interfere with the next stimulus, right? So cleansing your palate's often like just trying to get the previous stimulation off of the tongue, right? And wipe away that adaptation. All right. We can have other chemicals that interact with transduction mechanisms. So there's a chemical in toothpaste called sodium lauryl sulfate. And have you ever brushed your teeth and then drunk orange juice or soda? And it has a really bitter component to it. And that's because this sodium lauryl sulfate in toothpaste inhibits sweet transduction and sort of enhances bitter transduction. And we're going to see evidence of this today in lab because we're going to try miracle berries and Gemini tea. Has anyone ever done those two? Because both of those modify your transduction mechanisms and change the way foods taste. All right. Lastly, today, I want to talk about conditioned taste aversion, and then maybe we can get started in lab early, since it's a big lab today. Trying to get their list the whole time. So CTAs, conditioned taste aversions. This happens when we have a really negative event after a neutral or even positive stimulus, and usually that negative event is getting sick, right? And we have a strong, and then that neutral and positive stimulus takes on that negative value that it didn't have previously because we get sick, right? Lesion studies have shown that the PBN and the amygdala are the structures that are responsible for this. So if I go into a rat and I take out its PBN or I take out its amygdala, then that rat can no longer form a taste aversion. We think, and I'll talk more about this on Thursday when I talk about top-down influences on taste, but we also think not just aversions, but cravings also are generated here. So this is more, it's kind of like, the amygdala was first identified because fear was there, but it's actually all emotions. This fear was sort of the most pronounced one. Same thing here, we think that this is changes in taste. It's just aversions or a very strong change in taste and permanent, more permanent compared to cravings or come and go. All right, so taste aversion. You eat a bunch of cheeseburgers, cheeseburger tastes good, you like the cheeseburger, right? You get the flu, you get sick, right? And then after that, the cheeseburgers, even maybe the smell of cheeseburgers or the thought of cheeseburgers makes you start to feel sick. You definitely don't want to eat them. And if you do eat them, they don't taste good to you, right? So some switch got flipped in a single trial, right? A single pairing. Some switch got flipped in your brain that now changes the way that food tastes. Does anyone have taste aversion in here? Wants to share what it is? Everyone should. What are you averse to?
The cookies with M&Ms in them.
Okay.
Yeah.
Cookies with M&Ms, because you ate some and got sick afterwards. Yeah? Great.
Like the yum sauce at bocce places used to be my favorite food. And I threw it up and I can't be around it anymore.
All right.
I couldn't eat or even smell tater tots
Tater tots.
for like 6 years.
Oh, but you can get over it.
Yes, I got over it like 6 months ago.
But it takes a long time. You have to force yourself to eat it.
It was really bad.
Yeah.
So I was about to say, I don't have this anymore, but when I was like 2 or something, I was eating like more hot dogs than I could remember. So I couldn't eat hot dogs until I was like 10.
Yeah. Hot dogs, dude.
Tequila.
Tequila. Yeah. Tequila's got a really strong taste and smell, and it can actually make you sick. Like your body's actually doing what evolution said. If you consume something that makes you sick, don't ever do that again.
Mine's like anything bubblegum flavored or scented.
All the flavors of yeah, that's sad.
Mine's barbecue, but I didn't eat it for 6 years and then I had it again 3 months ago and I got sick again.
Oh, that's the worst. So if it happens more, I mean, it's single trial learning, but if it happens multiple times, then it's really hard to cause extinction, right? Cause extinction. So, all of those cases, right? You all cognitively know that that food didn't make you sick, right? And then if you eat that food again, it's not going to probably make you sick. But even though you cognitively know that, this is at such a subconscious level, it's hard to overcome that, right? It's hard to overcome that. All right, so again, the more times it happens, right, the stronger that becomes, and usually it doesn't happen more than once, unless you're in a situation where you're getting sick a lot, like morning sickness. Sometimes people develop conditioned taste aversions because they're repeatedly getting sick with pregnancy, or chemotherapy, which can make you nauseous, right? And it's worse in kids because they don't have as much cognitive capacity to understand that food's not making them, it's what's making them sick, it's chemotherapy. And so they actually have developed this like weird flavored, I don't even know how to describe it, flavored lollipop that they'll give to kids before chemotherapy so that they form a taste aversion to the lollipop and not to like normal food that they might have eaten that day or the day before or something to try to help them. Because this can be a serious problem where people quit eating during chemotherapy when they need nutrients and they need to stay healthy because of the way food has become a negative taste for them. All right, CTAs can be used as a research tool. We use them a lot in the lab. In fact, that was the very first experiment that I ever did when I came to Wofford was a conditioned taste aversion 2-bottle preference test. Don't even need any equipment. All you need is some lithium chloride in a syringe and 2 bottles. And we published it. And we did, we showed that rats could form conditioned taste aversions to free fatty acids, the fat molecules, right? So if you want to tell if a rat can taste something, right? If it can't taste something, it can't form a conditioned taste aversion to it, right? But if it can taste something, then you can condition it to taste aversion. The other thing you can use CTAs as a tool for is, does something make you sick? And when we talk about the vestibular system at the end of the class, which I was looking at my notes, we're like, I know we just had midterms, so I feel like the end is going to come like running out. So at the end of the class, the vestibular system, I'll talk about some research that I did in grad school where we showed that high-power magnetic fields, very intense magnetic fields can make rats feel sick. Because we gave the rats sucrose and then we put them in these high magnetic fields, and then the next day they formed a taste aversion, they had showed a taste aversion to the sucrose, right? So we can use CTAs in order to identify if you can taste something or if something makes you sick. All right, that's that. Trying to slow down a little bit.